So it's basically taking and looking at innate inborn defenses that you and I have available to us, and that's shown here as the first two lines of defense in the slide. And then this third line of defense is basically chapter 15. And I sort of liken it to the metaphor of you are a queen or a king in a castle, and you have to protect yourself from the invading Huns or whatever. And so in your castle, you have various lines of defense to protect you against these invaders, okay? And so this chapter talks about the first two lines of defense, and if the invader gets beyond those two, you have a third line of defense, and that's this specific immunity that we're going to get into next week. So I think you got to sort of make metaphors, analogies. You have to tell a story, as silly as sometimes the players of the story are. You gotta sort of see the forest through the trees, sort of thing. You don't get caught up, although it's important to get knowledgeable about the technicalities of all of this stuff. But you got to understand the big picture. Don't lose sight of the big picture here, right? You're trying to provide protection, and some of this you get naturally. You get it via your genetics. You get it via evolutionary processes that have developed in Homo sapiens over hundreds of thousands of years, right? So this is just another schematic that gives you a visual sort of a flow chart, same as the previous slide. And I always think it's good to be able to come back to these sorts of things to, again, see that big picture, right? Because sometimes you can get muddled down in the details. So if you get confused and you're like, where am I in this whole thing? Just take a step back, go to one of these diagrams maybe, and that may help get you back on track, so to speak. So we are basically over here in the left-hand 2/3 of the page in this chapter. And then again, this is the next chapter. So the chapter, in essence, just literally goes through these two lines of defense, starting with physical barriers and working all the way down to complement, and that's the end of the chapter. That's basically how it's laid out. So again, first line, barriers, physical barriers that we have at our disposal to protect us. A lot of this is just intuitive, should be perfectly understandable, right? We talk about it in A&P, the skin, one of the most effective barriers, even though it's only, you know, a few hundredths of a millimeter in thickness, your epidermis and dermis. And by the way, if you think back to A&P, the bloodstream, the dermal capillaries, are in those dermal papillae, which are right in the first layer of the dermis. So you have the epidermis, of course, above that before you hit the space, your you know, the room space, if you will, the outside of the body. So what I often tell my A&P students is when you think of the skin, yes, it's an effective barrier. The epidermis is stratified squamous, right? But we're talking about a barrier that is literally less than a few tenths of a millimeter thick. It's effective, but all you got to do is cut that with a knife or with a piece of paper. We've all had paper cuts. What happens to you? You bleed. Well, when you bleed, obviously that is a potential portal of entry, isn't it? Which this is described in the chapter as well. So yes, the skin is an important barrier, but it's a thin barrier. And then we have some other factors here, you know, the lysozymes from sweat glands. We have even the tears themselves have antiseptic properties, the fluid. So it's good to cry sometimes, right? Well, no, it's not good to cry necessarily, but you have in your tears antimicrobial chemicals. That's kind of cool. Stomach acid, if you swallow a lot of those bacterium that are in the food we eat, right? And really, we're not eating sterile food, right? Lots of our food have acceptable numbers of microbes in them. Think of milk, for example, right? It's pasteurized to lower the number of microbes, but you know there's an expiration date beyond which it's probably not safe to eat. So we ingest food that has bacteria in it, viruses in it probably. But the stomach acid, which is pH what? Give me a number. What do you think? pH, stomach, juice, gastric juice.
9.
No.
Oh wait, no. 5,
Dose.
4.
2.
Dang,
2.
that is very acidic.
Yeah, extremely acidic. Yeah. You know that scale goes from 0 to 14, 7 being neutral. So we're only 2 integers from the most acidic scenario possible. You can't create you cannot create a more acidic solution than pH 0. And I can't even think of any naturally occurring thing that would be that low, but theoretically you could make it in the laboratory. So pH 2, that's what you got in your stomach contents. Very acidic. And that's there, not the least of which, to provide protection to you. Yeah. And so we talk about the pseudostratified cilia, the columnar of the trachea and the nasal cavity, helping to trap any mucus that gets caught on the or bacteria that gets caught on the mucus, right? And what do the cilia do?
Catch particulates.
The mucus catches the particulates.
Oh.
So there's going to be mucus lining these cells. You're looking at the cilia here on the surface of the simple or the pseudostratified columnar cells. That's what are shown here in this photograph. But what are the cilia there for? They don't really trap them. They don't trap the particulates. The mucus does.
They beat the mucus and send it to the stomach.
Well, if it's in the trachea, the cilia are going to move the mucus upward, right? And then, Vera, you were thinking, we got to get it to the stomach somehow, don't we? Yeah.
Yeah.
So typically, if it's in the trachea, the mucus will be pulled upward, and then once it gets to the pharynx, you'd swallow it and then dump it down into the esophagus, which lies posterior to the trachea, right? You see the diagram here? This is the esophagus right here. It's not labeled. Well, actually it is. Well, the epiglottis is, but the esophagus is this. So the cilia are there just to move the mucus, eventually making its way either down into the stomach, or you could, I guess, spit it out too. And this is, again, is just reviewing some chemicals that we have at our disposal that have antimicrobial action. Interesting to note that semen has antimicrobial chemicals in it, but you didn't hear about that in reproductive system in A&P 2. I don't remember teaching that. We've all often heard about the vagina being very acidic and how that acidity helps to keep the pathogens at bay. But when you take, for example, antibiotic, what does that do to the lactobacilli, potentially?
Decreases it.
Kills it.
Right, and then what happens to the pH?
Drops.
No,
Increases.
it goes up. These guys help maintain the acidity. Okay, so if you take them out of the equation or reduce their number, then the pH tends to rise. And that rise in pH allows those opportunistic bacteria to take and gain a foothold, and pretty much what are you often dealing with at that point?
Infection.
What kind?
Yeast.
Yeast.
Right, yeah. So that is there to protect you. And that's not necessarily good for sperm cells, because, you know, when sperm is ejaculated into the vagina, a lot of those cells die because of the acidity, but enough survive and get beyond the vagina that they can, of course, get into the uterus and find their way up the fallopian tubes and maybe fertilize an egg. In addition to those chemical first-line barriers, we also have genetic defenses that protect us against microorganisms. That's what this is talking about. So something that might give your dog or cat an illness, a bacteria, a virus, you might be immune from that because of your genetics basically providing you with protection. As we are seeing with the COVID-19, one hypothesis as to how it got to Wuhan there in that market is that the virus may have made a leap right from an animal to the person. And that happens quite often in viruses. They jump from swine or often birds to humans. Those are the two major sources of most flu viruses that ravage our Earth each year. They come from swine or birds. So in that particular instance, the bird or the pig may not suffer ill effects of the pathogen because of its genetic protection, but when it gets to another species like me and you, we can get sick because our immune system has never seen that. While it may have, evolved with the pig or with the bird line for a long, long time. Okay, any questions on that stuff? Okay, we talked about this Tuesday, the PAMPs and the PRRs. Any questions on this? The important thing to understand, the takeaway message here is that our immune cells are able to distinguish the good guys from the bad guys. That's a fundamental tenet of immunology, the ability of immune cells. And when I say immune cells, I'm talking about white cells. They do this via molecular recognition systems. This is why in A&P, when we talk about the cell membrane and the fluid mosaic model that described the phospholipid bilayer and embedded proteins, remember this when you had it in A&P 1 a long time ago? Seems like forever now. It's so important to understand that, because how can you possibly understand this if you don't have some fundamental knowledge of what a cell membrane is made of, right? That's just critical. Because some of those embedded proteins in that phospholipid bilayer of the leukocyte are acting as receptors. They're acting as pathogen recognition receptors. Those are the eyes of the white cell. I'm not sure you're going to see this in quotes in your book. I probably made it up myself, but it helps me better visually make a story and understand this whole thing, right? So don't be afraid to think about that sort of stuff to help you better understand it. Now, when you go to talk about it on a test, don't just say the eyes of the white cell recognize, talk about the PRRs, and you can put that in there if you want, but we want to also not forget to use the right terminology. Okay. And those PAMPs are those surface molecules typically. They're typically proteins or glycoproteins that are what tells the white cell this is a foreign entity that probably needs to be neutralized. Okay. Good with that?
Yep.
Okay. You should watch this video. I talked about it Tuesday. These toll-like receptors are just an example of some of these PRRs. That's all they are. This is depicting the cell membrane of a leukocyte. Here's the cytoplasm, here's the outside. This video will talk a little bit about how some of these pathogen recognition receptors function. It's fascinating. A lot of them are based upon, of course, interaction with the PAMP, but also it talks in this video about how certain genes within the cell are turned on, and those genes, when expressed, produce proteins, and those proteins are going to help neutralize the invader. Would those genes have gotten turned on if there hadn't been a pathogen?
No.
No. It's the presence of the pathogen that sets into motion the activation of certain genes in the leukocyte. Now, again, you know what a gene is. We talked about that already, right? Everything builds. Segment of DNA that codes for a protein. At least that's one type of gene. This talks about the various systems we have at our disposal, many of whom harbor these white cells, these leukocytes that are going to help fight off potential foreign invaders. This simply reviews the formed elements, focusing in yellow on the white cells because those are the soldiers, if you will, that are helping fight the war for us. And this simply reviews the differentiation of cells that originate here in this hematopoietic stem cell and bone marrow. Remember this from ANP1. You should have had it in ANP1. And how that differentiates into two major types of stem cells, the myeloid and the lymphoid. Now, I don't want you to memorize all these intermediary cell types. It's not important, but just know that you have some precursor bone marrow stem cell from which all existing formed elements are formed. When I say formed elements, those are white cells, platelets, and red cells. Again, ANP1. These cells down here, you probably have never heard of. The dendritic cell here, I suspect you've never heard of that cell type before, but we will talk more about it in the next chapter. But it is basically derived from a monocyte, or it mentions your relatives of macrophages. What is a macrophage compared to a monocyte? Well, we often define macrophage as a monocyte that has left the bloodstream and gone out into the tissues. So it appears that when this monocyte undergoes diapedesis, which is what? Remember that from Tuesday?
Yes.
And that's the term that our friend, what's his name on the video, mispronounces, by the way. Drives me nuts. Diapedesis was the movement of the white cell between the simple squamous cells of the capillary. Remember that? Yeah. So when monocytes leave the bloodstream and go out into the tissues, it looks, just based upon the diagram here, the sketch, that there is a morphologic change. The shape of the cell appears to really change, doesn't it here? And so when we talk about macrophage, that means, you know, very large hungry cells. These are derived from monocytes. They're not a sixth type of leukocyte. They're just a monocyte derived cell. And then we will talk more about these dendritic cells more next chapter. But these can, like the macrophage, provide a real important function in removing pathogen by phagocytizing it. These natural killer cells, interesting, and I don't know if you can see this, my little pop-up thing is blocking this. It says, related to T cells, but display no specificity, active against cancerous and virally infected cells. So what this is basically saying when it says displaying no specificity is it doesn't act like the T or B cell does, which is able to respond to a specific foreign invader. Okay, it responds to a specific antigen. This natural killer cell is like an all-encompassing cell that will attack any and all foreign invaders. It doesn't necessarily target a particular type. Natural killer cells, they're called. Don't you love that name, natural killer? I'm glad I got those. This is obviously just a table that sort of goes over the 5 types of white cells, their features, their prevalence within the circulatory system. Again, neutrophils being the most common, basophils being the least abundant. I don't think you need to memorize this table or anything like that, but I think it's worth taking a few moments and just looking it over. I'm not going to ask you to identify a particular type of white cell on the test or anything like that. But we will be talking a lot about lymphocytes in chapter 15 next week. And please stop me as I'm going through this if you have questions. Okay, then we kind of shift our gears a bit and talk about the lymphatic system. Like the circulatory system, this is one of the primary highways through our body with regard to how potential pathogens can move, right? There's two major highway systems, the circulatory system and the lymphatic system. If you had to describe the lymphatic system, boy, it's a hard system to sort of explain, isn't it? Basically, what it does is it returns extracellular fluid back into the circulatory system. That's one function. There's multiple functions, but one of the major things that it does is it returns fluid that would normally be initially between the cells of the tissues. We call it interstitial or extracellular fluid. And we've got to be able to return that back into the bloodstream where it becomes part of the plasma. So when you look at the anatomy of the lymphatic system, we really have to start here at the lymphatic vessel, the capillary really of the lymphatic system. So here we see the red arrows indicating the movement of the fluid. Well, again, think of it as extracellular, or sometimes it's called interstitial fluid. And it's picked up by the lymphatic capillaries, shown here in green. And then you might remember from A&P, there are valves in these lymphatic vessels to allow unidirectional flow of the lymph. Now it's called lymph. Once it's in the lymphatic capillary, we call it lymph, L-Y-M-P-H. That's the name of the fluid. And it's very similar to plasma, blood plasma, although don't confuse the two. Blood plasma is not in the lymphatic system, and there's no lymph in blood, okay? So this lymph that's picked up by the lymphatic capillaries, which is typically near blood capillary beds, okay? So here's the capillary bed of blood, you can see that. It's kind of purple zone where we transition from blue to red. Blue is the vein, red is the arteries, right? As this fluid is passing through our lymphatic system, it's also passing through lymph nodes. And we talked about what they did the other day, right? What's the purpose of lymph nodes? Why do you have lymph nodes? Nadia, any idea? Nadia, are you there?
Yeah, I'm here.
Okay, just want to make sure you're alive.
Yeah, I had to plug my computer in.
Sometimes I see people appear, but I don't know that they're really there. In A&P the other day, I suspected there was a student that logged on just for the fun of it and just kind of was there to make me think she was there. And when I kept asking, no one ever replied. So anyway, any idea what the lymph nodes are here for?
They contain lymphocytes that fight off infections.
Right, there are special white cells embedded here within the node, and they are there to provide surveillance of the lymph. Right? It's basically what they're doing. And if we see a bacterium coming through in the lymph fluid, those white cells can presumably take action and neutralize, kill that pathogen. Go back, if you need to, into your A&P1 notes, if you still got them, and maybe take a moment and review that, like I said, Tuesday. In addition to the lymph nodes, here are some other additional lymphoid organs. which again, this is nothing new, although this miscellaneous section may be something you haven't, excuse me, heard of. It's just other lymphatic tissue spread throughout the body. In essence, there's several different examples. I give you a couple here. And then we get into basically the second half of the chapter, which focuses on the second line of defense that provides us with protection. Again, I think this is fairly straightforward. Are there questions on the inflammatory response? Here's diapedesis. We talked about that a couple moments ago. Did anybody try this hyperlink to see if it played a video? No. I'm going to try it. I'm not sure it's going to play. If it does, we'll watch it. I went through this chapter the other day, and I thought I put an additional link down below. I'm not sure why it's not showing. Let's just try and see if it works.
It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
I think that is a really nice video, and you might need to watch it 2 or 3 times, but it does, I think, a very good job describing numerous components of the inflammatory response. I mean, there's a lot of stuff there. But when you think of inflammation, see if I can get out of this.
The inflammatory response.
When you think of inflammation, what do you think of?
Swelling.
Swelling. Mm-hmm. You've all suffered inflammation. What do you suffer? What do you feel?
Pain.
Pain. Tenderness. Maybe the area looked red, right? These are all characteristic inflammatory symptoms. This video goes a long way to explain what the swelling is due to, right? Which they described as the permeability of the capillary bed has changed and fluid begins to leak out. Of course, the neutrophils and other white cells also undergo diapedesis. So that's where a lot of the swelling occurs as a result of that response of the blood vessel. Chemotaxis is the same thing as, you know, pouring buckets of blood out the back of the boat if you want to catch a shark. Right? The chopped up fish that you had in the bucket of blood that you dumped over the back of the boat attracts the shark. The shark has amazing olfactory reception skills, and it follows the line of blood to the boat, and you throw out a big hook with a big mackerel attached to it, and you catch the shark. Well, this is the attraction of what sharks? What are the sharks here?
The white blood cells.
Right. And the release of the blood, the chum we call it, or the buckets, that's chemicals given off by the damaged tissue that attracts the phagocytes. It could be given off, as I said, by the damaged tissue. It could be given off by the pathogen. There's a number of different chemotactic, there are different types of chemicals, I should say, that induce chemotaxis. But this is a really critical part of the process. Questions on that? Does this make sense? Are you okay with it? Are you not okay with it? I'm not getting any feedback, so I don't know where you guys are. Anybody? You ready to move on?
Yeah, I'm okay so far.
Okay. Again, I think this is pretty self-explanatory. Fever, we'll let you look that over. Phagocytosis, you know what that basically is. The video talked about the neutrophils and other white cells like macrophages, basically gobbling up the pathogen. This is that unique ability of a white cell to recognize via the PRR and the PAMPs, right? That whole thing.
Mhm.
Here are 3 of the most phagocytic types of white cells. It kind of depends upon what the offender, if you will, the pathogen is. For example, the eosinophil, we tend to talk more about it when we're dealing with parasitic sorts of infections. Parasitic being things like trypanosoma, plasmodium, giardia. Remember us talking about that like the second lab, and there was a little section in the eukaryotic chapter we talked about some protozoa that can cause problems. And so the eosinophil count in somebody who's suffering a parasitic infection, for example, would be elevated. So when you have a CBC done, a blood draw and a blood analysis, and it comes back from the lab the next day, the physician can look at that and say, oh, you have an elevated eosinophil count. Where have you been the last 3 weeks? And you say, oh, I've been into Brazil, went on down the Amazon. Well, okay, I bet I know what's going on there, right? So they connect the dots, so to speak, and can often try to narrow it down based upon things like blood analyses. If you're sick in bed and you get a blood draw, you're going to have an elevated level of maybe neutrophils, indicating that you are suffering from an infection. So that's kind of what this gets at. This is talking a little bit more about how white cells have those surface pathogen recognition receptors, PRRs. This is just one example of a PRR, which the video that I asked you to watch will talk about. So this is the eyes of the macrophage, right? This is what's going to bond with, if you will, the foreign invader. It could be part of the cell membrane or cell wall, I should say, of the bacterium. It could be a toxin that the bacterium releases that interacts with this receptor. We call it an antigen, and we'll talk more about antigens in the next chapter, but it's viewed as a foreign substance. So it's really critical that the white cell be able to identify self cells from non-self cells. Okay, and this is exactly what I was talking about earlier. This is what the video will talk about. And when that foreign molecule combines with the pathogen receptor, or the I always forget the name of it, the pathogen-associated molecular, or I'm sorry, the toll-like receptor, pattern recognition receptor of the white cell, that can set into motion, as I said earlier, activation of genes, which results in certain chemicals being given off, in this case by the macrophage, that can kill the foreign invader. It can also promote the inflammatory response even more. In other words, call in more troops. This just talks a little bit more about how the macrophage, once it's phagocytized the foreign invader, the bacterium in this case, it talks about how it kills it through this respiratory burst. Very interesting. The book talks a little bit about that, so I'll let you read that over. This is an internal phenomenon that occurs within the macrophage following phagocytosis. Okay, so here are the bacteria. They've combined with the toll-like receptor, or the PRR. It's brought in. It gets compartmentalized, right? The lysosomes produced by the cell, which contain powerful hydrolytic enzymes, basically digest the bacteria. And then they talk about this respiratory burst that does the bacteria in once and for all. Really interesting stuff. Interferon is another chemical that some white cells produce in response usually to viral infection. Interferon can also work against cancer cells as well. And I'll show you a little video here that talks about interferon. So let's see if we can watch this.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
Were you guys able to watch that video?
No.
It didn't come up on mine.
Okay. Did the previous video that I showed come up on your screen?
No, I thought it was just because my internet is so slow here that it just, I assumed everybody else could see it, but I couldn't. But I'll go back and watch them.
You guys couldn't see it either?
No.
Okay, okay.
I couldn't see the video.
Okay. I don't know if I should have, like, you know how we've had issues with some of these, like, Word documents that I open up, I have to reshare it?
Mm-hmm.
Yeah, I don't know if that's the case here. Which I'm just not sure if I can show you the video or not, I guess. But you can go in and watch it. And you may need to watch some of these 2 or 3 times. They're short, they're like 2 minutes long. So what the video talked about basically was you had this initial cell. Maybe it's a macrophage or a white cell of some sort, and the virus is basically brought into the cell. You have, or anyway, you have the nucleic acid well, if it's an envelope virus, it's going to be brought in, and this is going to result in the cell making more viral particles, right? You know how viruses work generally. The cell is taken over, the cell creates more viral nucleic acid, more viral capsids. The viruses are put together, and eventually they lyse from the host cell, eventually, right? So this cell can't do much upon being invaded by the virus. But what this virus also does, in addition to making more viruses, is that it has one final trick up its sleeve, and that is it will produce IFN, that's interferon, because it's been invaded by the virus. So it cannot save itself, like the video said, but what it can do is it can release interferon in response to having been invaded by a virus. What the interferon then does is it can attach to special protein receptors on an adjacent, another different white cell. And what that does is that turns on certain genes within this white cell that will prevent the virus, which would presumably like to target this cell too, right? It prevents the virus from replicating itself. So this cell is protected by having had the interferon induce these cellular changes that result in the virus having basically no effect. Does the virus get into this cell? Yes, but the cell's machinery is not taken over like it was in this cell. Does that make sense?
Mhm.
Okay. Yeah, it's really, really interesting. Again, interferon, when I think of interferon, I think of this occurring during viral infection and also helping to fight cancer. Sort of a built-in innate system that we have at our disposal to try to protect us from viruses and cancer. And this last topic of the chapter, I believe it is toward the end of the chapter, yeah, is talking about something called the complement system. Now, this is probably one of the more difficult concepts to understand, at least for me it is. This first bullet just kind of describes the fact that you have a bunch of these special types of proteins that are circulating around in your body, in your bloodstream, perhaps even in the lymphatic system. I would think anywhere you could have viruses and bacteria and pathogens, you've got these complement proteins that are just floating around. And if you look at the word complement, like somebody pays you a compliment, it's something nice, something beneficial. Well, this is a beneficial process that helps to protect you. Whether that is in innate defense against any and all pathogens, or whether it's when you fight a specific enemy, which we're going to talk about specific immunity in chapter 15. In either of those two scenarios, these complement proteins are at work. Okay. And you should watch this video. I don't know that I can show it to you, but you should watch this video. It's probably like 2 or 3 minutes long. And it's something you may have to watch several times. But basically what it does is it talks more about how these complement proteins work together to knock out pathogens. And they do this in a number of different ways. As the second bullet here talks about, these complement blood proteins, well, they specifically say blood, so maybe it's only in the circulatory system. I was thinking maybe it might be in the lymphatic, so it may not be there. So in the circulatory system, working in concert with inflammation and phagocytosis to destroy a number of different pathogens. How do they do that? Well, the video that I just showed you earlier that you couldn't see, talked a little bit about the white cells leaving the capillary bed, going out into the tissues, how chemotactic chemicals were given off, and that helped to encourage the white cell to move toward the infection site, right? Well, one way that some white cells know to gobble up the bad guys is via certain complement proteins coating the surface of the pathogen and making it more likely to be identified as a pathogen. So that's one way that complement proteins work. They basically coat, these proteins will coat the pathogen. It's called opsonization. It's sort of like, this is going to be a really stupid analogy, but maybe it'll help. I like peanut M&Ms. I like the chocolate around the peanut. So the chocolate coat, all right, the peanut is the bacterium. The chocolate around the peanut and that shiny yellow coat, that is the complement proteins that have opsonized the bacterium, covered it, coated it. And because it's coated, then I can eat it. The macrophage can identify that chocolate-coated bacterium as being something that should be eaten. So that's one way that complement proteins work. They coat the pathogen. They get around it and they coat it. And it makes it easier to recognize as a object that needs to be phagocytized. Your book on page 460 talks about another mechanism that that describes complement action. And at the top of that page, you see where it says initiation. If you have your book, flip to page 460. Give you a minute to grab it. Figure 14.20. At the top of that page, there's a yellow box, an orange box, and a blue box. These describe 3 different pathways of how complement proteins work. And I don't want you to necessarily know about the difference between those 3. I simply want to mention that where it says classical in yellow there, that is working during acquired specific immunity. That's the next chapter. That's targeting a specific pathogen. Those other 2 pathways, the one in orange and in blue, the MB lectin and alternative, those 2 pathways are involved in non-specific innate immunity. Basically meaning that no matter how many times you've been exposed to any pathogen, no matter what pathogen it is, these 2 pathways will kick in and result in the demise of the pathogen, basically. So one is very, very super specific to a given pathogen. The other 2 are more general. That's all that means. Yet all 3 pathways will ultimately cause holes to be formed in the cell membrane of the bacterium in this particular example. Okay, and that's what's talked about here in number 1 in this slide where it says the membrane attack complex. Because if you look at that figure toward the bottom where it says membrane attack and polymerization, steps C and D, in the video that I alluded to a moment ago, the YouTube video that you're going to be watching, we'll talk about this. Do you see where it says C9 and C5 and C8 and C6? See that in the diagram? Part C? Yeah.
Yep.
C stands for complement, And then each of these complement proteins is given a number, basically. And so some of these particular proteins, these complement proteins, have to work in a particular sequence. And in this particular example, it's the C9 complement proteins in blue that actually form these little holes or pores in the membrane of the pathogen. And that membrane attack complex, it's called, the MAC, that formed as a result of C5 and C6 initially starting that whole process out of pore formation, if you will. So these complement proteins work in a particular coordinated manner that ultimately cause pores to form in the cell membrane. And of course, what's going to happen to the cell contents? They're going to leak out and the cell's going to die. Yeah. So isn't that kind of interesting how these proteins, these blood proteins, can become activated, and one important way that they kill bacteria is to poke holes in their cell membrane or really form pores in their cell membrane, and then the contents leak out, cell dies. So it doesn't make any difference whether it's innate or specific. Both of these, or all three of these pathways ultimately lead to these membrane attack complexes being formed in the pathogen. So don't get caught up in all the technicalities, but sort of get, again, the big picture here. And watch that video that I mentioned in one of the earlier, like the second slide before this. This second part here of complement, this is what I was talking about before, the peanut M&M, right? The opsonization, these complement proteins will coat the pathogen, and then the macrophage will say, oh, yellow peanut M&M, let's eat that. If the peanut had not been coated by chocolate, it's possible the macrophage may not identify it or be as likely to phagocytize it. Having that extra chocolate was important. Again, silly, dumb story or analogy, but if it helps you kind of picture this, then do that. And number three is just, I think, getting at the fact that it amplifies the inflammatory response. These complement proteins can kind of fire up the inflammation, which in the long run is good, even though we don't like to feel tenderness and pain and swelling. That simply means that our immune system has identified, the fact that we have an infection of some sort, and we're going to send phagocytes there to remove those potential pathogens that are causing the infection. This is that same video, I believe, that I had hyperlinked in the YouTube several before, and you should be able to watch that. And then that's basically leading us to chapter 15.